Hello back. Now we're talking about the fundamentals of our nervous system. The nervous system connects us to the external and to the internal environment. It's the master communication system of our body, which controls and coordinates everything that happens in the body. It is made up of a computer-like thing, which is the brain and the spinal cord, and we call that the central nervous system. And then we have cables that connect it to the body. And we know that as the peripheral nerves or the peripheral nervous system. The central nervous system, we often call it CNS, the peripheral nervous system PNS. When we look at the basic functioning, we're going back to the whole thing of the negative feedback loop. It's the same thing. We have a receptor that detects a change and sends a sensory impulse to the brain or the spinal cord for interpretation. After that impulse is integrated and the information is integrated and we know what to do about it, we'll send out a motor impulse to an effector, which is either a nerve, a muscle, or a gland, and that will then elicit a change. Nervous tissue is reactive and needs to be stimulated. We use terms for that quality that is excitability or irritability, a responsiveness. Irritability doesn't mean somebody's pissy, it just means when you poke it, when you stimulate that nerve, it will react. The nervous system communicates by electrical impulses through wire-like extensions. And this makes it very fast and very specific. Compare that to the endocrine system, where one gland drips a few drops into the bloodstream and then somebody else, the target cell, picks up that chemical. The nerves are cables, and the cables send an electrical impulse from one side to the next side, from a beginning point to an end point, and at an end point it will release a chemical and act specifically on that target. So it's like the electrical system. Electricity, when we look at that, since it's an electrical system, electricity is based on the flow of positive and negative charges. In our body, those positive and negative charges are ions. They're charged molecules and they can flow. Remember the ionic bond. One atom steals or donates an electron to another atom, and that makes both of these atoms charged. One is positive charged, one is negative charged. And those are then our ions in the body. Also our electrolytes that we then drink, for example. One concept in electricity that's very important is that opposites attract and likes repel. Magnets repel or snap together, depending on if you put the same pole towards themselves or if you have an opposite pole that goes to one magnet. So the same will repel, the opposite will attract. That's the concept here. Opposites attract. Ions work the same way. If we have areas or fields of positive and negative charges, ions will tend to move towards the area of opposite charge. So if an area is more positively charged, a negative ion will move towards that. A positive ion will be repelled from that. A boundary maintains the different charged fields. In our electrical wires, we usually have plastic around them. We don't expect to touch the wire unless there is a plastic around it because we need that insulator. In the cells in the body, it's a plasma membrane, and that establishes an electrochemical boundary because, ions are hydrophilic and they are not able to cross a hydrophobic part of the membrane, the phospholipid bilayer, unless there is a channel. If there's a channel, they can just go through because we don't have to penetrate oil. The other way we can have a pump, and the pump also pumps these ions to the other side without having to go through the oil film. Gated protein channels let ions cross the membrane carrying a charge with it when they open. So, when you have to bring one charge from the outside of the cell to the inside of the cell, so we bring ions from one side to the other side, we gotta have channels that help us do so. When those channels are closed, nothing penetrates through. When those channels are open, these ions can move freely through it. When we look at how ions move, they always move, or molecules, they always move from a high concentration to a low concentration, unless we have it be an active movement, like a pump, where we move against that gradient. But then we need ATP. That's the difference between active and passive transport. Remember, ATP is needed in active transports, and passive transports use the chemical gradient to do the movement. Now, how it works here is, you look at the bottom left picture, the pump will establish more sodium. We use sodium a lot in this situation as well as potassium. So the pumps will establish more sodium on the outside of the cell membrane. On the outside, on one side of the cell membrane, but not inside the cell, on the outside of the cell. Potassium will be established in a higher concentration on the inside of the cell. That means a lot of potassium inside, very, very few outside. The establishment of those chemical changes, those chemicals being pushed to one side, that's established by the pump, the sodium-potassium pump. And now when a channel opens that lets sodium go through it, of course, there's much more sodium on the outside, so sodium wants to rush to the inside. And so that's a chemical gradient that the sodium is going to rush inward. What happens though, since the sodium has a positive charge with it, by doing so, it will also bring that charge from the outside to the inside of the cell. And so the ions move because of a chemical gradient, but it will carry with it a charge, and that will make a difference in the electricity. Now we get to the concept of voltage. Voltage is that charge difference between two charged fields. So now we can take an outside of a cell membrane and the inside of a cell membrane, and if there's a difference in charge, a difference in positive and negatives, we have a voltage. And reactive cells respond to a change in voltage. If the voltage changes sufficiently, that means the channels open, the outside is more positive, the inside is more negative. So we open a sodium channel, we let a lot of sodiums come in, they carry a charge with it, the positive charge. They bring that positive charge to the negative end of the field, the inside of the cell. And that, as a net effect, will make the inside less negative, or we could call it a little bit more positive. And so the voltage, therefore, will be reduced. And so the polarity, the difference between those two sides, the outside and the inside, is becoming less. And in technical terms, we call that voltage change there, we call that a depolarization. We make it less polar. A nerve impulse is exactly that. So we have ions that move across the cell membrane that changes a voltage, and then the cell reacts to that change in voltage. An electrically reactive cell then needs an established voltage at rest, and we call that a resting membrane potential. That's the difference in charge between the outside of the cell membrane and the inside of the cell membrane. In a cell, the resting membrane potential is approximately 60 to 70 millivolts more negative on the inside as compared to the outside of the cell. And the establishment of that voltage is mainly accomplished by the sodium-potassium pump, because we're pushing chemicals from one side to the other side. Now I know you're going to tell me, well, sodium is positive and potassium is positive, so what's that going to do about it? I understand. The main inside of the cell has a lot of big, large molecules that make it more and more positive, negative on the inside. Those are, when you look at the top middle picture, those are these A negatives. Those are these big anions, they call them. So they make the inside more negative as a whole. But right around the cell membrane surface, the charges that come in and out will make a lot of that fluctuation happen locally. And so when I then bring there a lot of sodiums from the outside to the inside in that local space of that cell membrane area, that will change the voltage. As well, then I can take potassium and push them to the outside of the cell, and that also will change that voltage. The sodium's coming in, brings positives in, potassium's going out, brings positive back out. One makes the inside more positive or less negative compared to the outside, and the other one, the potassium going out, will make the inside again, we take a positive away, so we make the inside more negative again. So let's do that again. Ions cross membrane shift voltage. Sodium are at a higher concentration in the extracellular space, rushes in when the channels open, the channel proteins open. So that carries that positive charge inside, changing the voltage on the inside, making it react when a level is reached that is going to be determined, a level of changing that polarity between the outside and the inside. So that's depolarization. And then potassium is kept at a higher concentration on the inside of the cell and opening its protein channels. They're different ones, they're not the same than the sodium ones. That will then carry a positive charge to the outside, will lower the voltage again on the inside. Will make it more negative again on the inside compared to the outside. So that will make the polarity greater again. And we call that repolarization.